Good morning, and thank you all for coming. I'm Joe Kraft. I'm on the faculty at the university. I'm also the scientific director of the Colton Center for Autoimmunity at Yale. And this is one of 4 Colton centers at University of Pennsylvania, at NYU, and at Tel Aviv University. And you'll hear speakers from the first 3 this morning. And we work together, and our purpose primarily is to explore ideas in immunology, autoimmunity, and to try to develop intellectual property and commercialization. And that's an opportunity today is to have several of our speakers from our collaborative institutions present to you their work, which has moved along fairly quickly, I think, some of which has commercialization interest, some of which has commercialization already, but all of which would like to hear from you and get your input into what they're doing. So very briefly, we are the Colton Center for Autoimmunity at Yale, so we're one of the 4 centers. And we're also one of the 3 accelerator funds at Yale Ventures, along with the Blavatnik Fund, devoted to the life sciences, and the Roberts Fund, developed to engineering, medical engineering and engineering. We, of course, are focused on autoimmune diseases and allergic diseases, immunological diseases, broadly speaking. We were launched in 2020. All the 4 centers were launched at about the same time, roughly speaking. And over the course of the past 5 years, we provided over $5 million to investigators at Yale. We funded some 40 projects, all of whom have presented good science. But we're interested not only, of course, in good science, but also in commercialization potential. And that's a major criterion for funding. And the investigators you'll hear from today have passed that bar, not only at Yale, but at the other centers. So this is what we're about, and we work through Yale Ventures. I should introduce Makoto Yoshioka. He's the director of the Colton Center, and this is really his baby. He's really led the way. I focus on the science. Makoto focuses on the commercialization and really setting up this symposium, so he deserves all the kudos for that. And eventually, in addition to out-licensing, venture creation, and industry partnerships. And we look forward to your comments about that on the work today. We've had some success in terms of out-licensing and in terms of company development. These are just 3 examples where we've out-licensed ideas to Biohaven and have supported work that's led to 2 company development of 2 companies, which have already received Series A funding or are well on their way, I think, to developing viable products. This is our core team. I'll focus on the individuals at the bottom. Makoto and I are up here, along with Carrie Lucas, who is the Associate Scientific Director of the Colton Center for Autoimmunity. All the people at the bottom, you'll see some of these individuals walking around today. These are our student and postdoctoral associates. Again, this is a program set up by Makoto and is to provide training and administrative skills for graduate students at Yale and postdoctoral fellows at Yale who are interested in technology transfer, technology development, and the idea is to help them launch their careers in that space through working with us. So really, thanks to them for all the organization as well, working with Makoto. This is a large group of advisors. When we review projects, of course, we call on external expertise. And again, this has been set up by Makoto through Yale Ventures. We have a number of entrepreneurs and residents who provide critical information, critical feedback on our projects, help vet the projects so that our external advisory board for the Colton Center can make final decisions about funding. So without their expertise, I don't think we would be as successful as we have been. All their names are listed, as well as the names of the external advisors, some of whom are scientists at Yale, some of whom are from the commercialization sphere as well. And we're very grateful for them for what they've done. This are a recent retreat held last year. We have another retreat coming this year, at which all investigators from all 4 of the scientific centers, Penn, NYU, Tel Aviv, and Yale, meet together to exchange ideas, to talk about science, but also talk about intellectual property development. And it's really been a very important forum for all of us as we try to advance what we do. So this is my last slide. These are our speakers for today, listed in order. To facilitate, time is tight, of course, so to facilitate the speakers having enough time to present their work to you, but also have enough time to answer questions. They will introduce themselves. I'll help moderate the questions from the audience, so please have questions available, but they'll do the introductions. But I will introduce Taku Kambayashi from the University of Pennsylvania, who will speak first, who's traveled up from Philadelphia last night. Taku, thank you. We'll hear from 2 investigators from Penn, an investigator from NYU, and then 3 investigators from Yale. Taku, thank you.
Well, good morning, everyone. Thank you. I really thank Joe and Makoto for inviting me here. It's really a pleasure to meet everyone and be able to present my science and also my business plan. So today I'm going to be talking about a skin-based therapy that we've developed for the treatment of lupus nephritis, but also for other conditions like obesity and skin barrier disease. I am representing a company that I founded called Abrax, but also I'm a professor at the University of Pennsylvania. The discovery, the technology that Abrax is IP that was generated at the University of Pennsylvania through support of the Colton Center. So I just go over the team. This company was founded about 4 years ago with my college buddy from Johns Hopkins, Joe Narita. He's in business, so he's the business side of things. I'm on the science side. We've recruited a great team of experts on the business side, all of which have a scientific background and a business background. We have collaborators that are really important, really multidisciplinary because as you'll see, it's a multidisciplinary approach that we have. So we have Moshe, we have a nephrologist, we have a medicinal chemist, we have a dermatologist, cardiologist, endocrinologist, and of course legal. So this is our team. So what is it that we're trying to do here? So I'm going to try to change your thinking of how the skin, really, it serves a purpose. So you might think that the skin is just the barrier between us and the environment. It protects us from stuff getting in. But really, the skin does much more than that. The skin is, what we think, is an endocrine organ, right? So they produce substances that can affect systemic processes, right? So what I'm depicting here in this diagram is that we've discovered, for example, some of the triggers like vitamin D stimulation and other mediators can trigger keratinocytes. So these are the epithelial cells in your skin. And then they will produce skin-derived hormones. I'm going to describe, there's many of them. I'm not going to go through all of them, but they're skin-derived hormones that then can travel. So these skin-derived hormones are what travel through the bloodstream and then affect systemic processes. I'm listing a few here. All right, now there's anti-inflammatory effects that provides protection to the kidney. There's increased skin and eye barrier function, decreased lipid metabolism that then lead to all these benefits like treatment of lupus nephritis, which is the focus of the topic of my talk today, but also be good for treatment in eczema, atopic dermatitis, obesity, and other disorders that are listed here. So the topic of today is lupus nephritis. We think that we have great preclinical data to support the use of of our approach in treatment of the severe complication of systemic lupus erythematosus, or SLE. About 50% of patients who have SLE will develop eventually lupus nephritis. It's a big problem because 40% or so of the patients will then end up developing end-stage renal disease that requires dialysis, it requires kidney transplant. It's a huge economic burden to all these countries. And the existing therapies, while there are, right, have their own, there are some pluses and minuses. And some of the existing therapies include corticosteroids. Nobody wants to be on long-term steroids, but that's the first line. You have lots of immunosuppressants like MMFs, cyclophosphamide, and also more recently biologics like Rituxan and belimumab, which targets B cells. So this is the landscape. And this is a growing market. It has about a CAGR of about 11%. It's thought to reach about $5 billion in terms of market value in 2033. So where's the market opportunity? I would place lupus nephritis kind of at the verge of an orphan indication. So it's not extremely rare, but would be rare. So it would be defined as maybe the target population that I have outlined here. The prevalence about 1.5 million patients in the U.S. alone, about 5 million globally. And then of those, what we estimate is the cervical targeted, the market would be about 250,000. But if we subset that, maybe it'll go below 200,000, so we could get orphan indication if we get a drug approved in this area. So that of course provides benefits in terms of market exclusivity and tax benefits. So we think the beachhead market would be in the U.S. There's a high unmet need, even though it's a rare disease. Again, I told you this is a debilitating disease. And also in terms of our approach, we're not only targeting lupus nephritis, that's one thing, but we think this has a lot of platform potential. So what our solution is, so all the drugs that I talked to you about is they're oral, they're immunosuppressants, they're biologics, they're injectables, they're very costly. Our solution is a topical one, right? So we're going to utilize the skin to heal our body, right? So it's non-invasive. We're activating a skin pathway to have a dual anti-inflammatory and direct kidney protective effect. It's easy to use, it's low cost. Again, outlined here is just a summary of the mechanism. We're applying this drug called MC903. We formulated this new product called F275 for the company, and it activates keratinocytes, drives skin-derived hormones, and then has anti-inflammatory and kidney protection. Here is just some preclinical data showing you that it works. So this is a lupus nephritis model in mice that that are broken into 3 arms, one that were treated with a placebo, one that's treated with MC903, our drug, at the very start, but also importantly, after the disease develops. And as you can see, the blue circles are the ones that are placebo. You can see that the proteinuria, which is a marker for renal damage, goes up over time. But if you look at the red and the green, the red, always never goes up. The green goes up at week 5, but when we start the treatment at week 5, you can see that it comes down way back to baseline level. So we think that our drug not only can prevent lupus nephritis, but also can treat lupus nephritis. So I'm not going to go through this busy table, but there's all these drugs here. The problem here is that they're immunosuppressive, and they're costly. A lot of the biologics are very costly. We think we can differentiate ourselves from these currently available products. It's a topical, non-invasive approach. It's outpatient friendly. It's non-immunosuppressive. It's going to preserve immune defense. It's a novel mechanism, and again, it has platform potential. In terms of market observations, it's pretty fragmented. People are using different types of modalities. Maybe we could bring it together, and because there's a huge unmet need in outpatient for mild and moderate lupus nephritis. So where do we stand in terms of our company? We've gone through seeds 1 through 4, really small increments. But what we've completed so far is that we now have a product. So we have this lotion that contains MC903. It's really easy to use. It dries quickly. All we're asking for patients to do is put this drug on their arms once a day. It dries in 10 seconds. You're done. That's all we're asking, and that's going to serve its purpose. We've done strategic consultation. We've had a pre-IND meeting for a different indication, in this case, atopic dermatitis. We have preliminary preclinical safety, efficacy data, and we've also developed, importantly, a new chemical entity. So there are drawbacks to MC903. It is a vitamin D3 analog. There are certain side effects that are associated with it. We think we've actually developed a new chemical entity that will abrogate those side effects. So we're really excited about that. In terms of our next round, we are currently raising for seed 5, about $3.5 million. We've divided in 2 sections. One is $500,000 for development of future pipeline. So that's really about the development of the new chemical entity that we're really excited about, that will give us new IP, as well as really mitigate some of the concerns that we have with MC903. And then we want to fund a first-in-human pilot study. All the data we have so far is in mouse. Also with human cells and human cadaveric skin and stuff like that, but still we really need to go first in human. So we've already talked to, we have an Australian subsidiary called Abrax Australia, where we're doing planning clinical trial because there's a tax rebate and there's a lot of benefits of doing studies in Australia. And this is just a design that, and we're asking about $3 million for that. So the development roadmap, so we've finished early development, reformulation of MC903. We've tested prototype formulations. We're in step 2, where we've targeted lupus nephritis. We have a lot of substantial preclinical data to show its efficacy. We're trying to optimize these new chemical entities, and we're trying to develop a first-in-human clinical trial. And then we're going to be moving to step 3 and step 4, which will include other indications such as obesity and skin barrier diseases. And finally, just to summarize, just give you the highlights of the investment opportunity here. We feel that we have an expert SAB and collaborators that's really going to be important for this company because it is multidisciplinary. We're really covering large areas. We're covering skin, we're covering nephrology, endocrine. We need a medicinal chemist to develop and further refine our new chemical entity. We have proven science. We have lots of peer-reviewed publications in journals like Science. We have this global exclusive license from Penn, so the IP that we generate in a lab has an exclusive license to our company, and we're continuously developing new next generation skin-mediated therapies. And I guess I'll end there. Thank you.
So we have a few minutes for questions, please. Yeah, can you briefly tell us more about MC903, where it came from, mechanism of action in the skin, and most importantly, toxicity, both topically and systemically?
Right. So MC903 is an already FDA-approved drug. It's used for the treatment of psoriasis. It's called Dovanex. It's off patent. It's been used for 30 years. It stimulates vitamin D receptor. So what we're trying to do here is a 505(b)(2) pathway, so that does alleviate some of the regulatory hurdles, unless we go to our new chemical entity that we developed. In terms of toxicity, so the number one toxicity with vitamin D in general is hypercalcemia. That is largely mitigated with MC903 because MC903 doesn't bind to this protein called vitamin D binding protein that is required for it to get stored in the fat of the body. So it's cleared very quickly. It has about a circulation half-life of 5 minutes. Still, there is a very, very slight chance that you could get some hypercalcemia. The major side effect is really skin irritation. And that's because it stimulates a lot of some inflammatory genes in skin keratinocytes. It's relatively mild, and once you stop the treatment, it goes away very quickly. But that still is a side effect, and it occurs about 10% of patients. But we think that our new chemical energy actually completely abrogates that.
I see there's an animal model of atopic dermatitis.
That's right.
Using it
That's right.
topically.
That's right. And actually, our new chemical entity completely gets rid of that.
Okay.
Yeah, but still have the same effect.
Yes, please.
Can you tell us what these hormones are that make keratinocytes?
Yes, I can. So
Have you actually measured them?
yes, we have. So this is unpublished. It's okay. But yeah, so some of the, initially we thought, TSLP was one of them, we thought. But actually, we've later found out that TSLP is just inflammatory. It's not really important for our process anymore. We've used TSLP deficient mice. But what we found is actually the skin produces bile acids. So bile acids, you would think, are made in the liver, right? And they emulsify fats in the gut, and that's all they do. But actually, bile acids are hormones. They're cholesterol. They're like steroid hormones. They're cholesterol derived. And we found that again, unpublished, that the skin actually makes bile acids. We can measure it in the skin. We can measure the enzymes that lead to the production. We can see it elevated in circulation. We can see it elevated in the kidney. So we think it's bile acids. We have a lot of data. We can, anybody who's interested, I could show later on that really show that it's skin-derived bile acids. And there's lots of different kinds of bile acids. That's why I call it skin-derived hormones, because it's not like one particular one.
And how do they relieve nephropathy?
So there's two things that happen, we think. One is that we think it blocks mitosis of neutrophils through a receptor called FXR. So these are hormones. So they stimulate different types of receptors in immune cells and also kidney epithelial cells. And our collaborator, Moshe Levy, had shown that FXR agonists have a direct kidney renal protective effect in obesity-related kidney damage and diabetic nephropathy. And that is through downregulation of lipid biosynthesis in the kidney. And also affects, in some extent, the podocyte effacement process. through FXR agonism. But so there's that's why I was saying it was a dual anti-inflammatory and a direct kidney protective effect.
Thank you for the presentation. I was wondering the relative contribution of the skin versus other systemic sources of the same...
Of bile acids, you mean?
Or any other mechanism. So how do you plan to counter that with just a topical to the skin?
I'm sorry, maybe I misunderstood. Can you repeat that?
I'm not clear on whether the skin is the primary driver of disease. So, if you're just using your mechanism, is it enough, to be able to counter all the other systemic stimuli leading towards lupus nephritis?
Right. So I don't know if we could. So I mean, that's a good question. I don't feel that it's a cure. It's not really a cure for the disease. It's more about suppressing the inflammation. But it has been shown that if you can suppress the inflammation for more than 3 years, then the outcome for lupus nephritis patients is significantly higher. So there is a time frame while which we want to suppress that. And there's compliance issues. There's many reasons why people will flare up and come back. And if we can have a method where we could use that's very convenient to use, that patients wouldn't mind because there are hardly any side effects, right? Then I think we've really achieved.
Lupus nephritis is generally treated with a combination immunosuppressive, several drugs, and to have a drug that you can add on and potentially down modulate the doses of the other drugs would be a benefit, presumably, but if it doesn't cause too much immunosuppression. It's something that's easy to apply. Lupus also affects the skin. It's the most common manifestation.
Yep.
So having a topical agent as another primary benefit. Okay.
Okay. Yeah. All right. Thanks so much.
Will be around. So please find them after our session and ask questions if you have any other issues.
And just a reminder to everyone walking into the room, thank you for coming. There is an overflow room, so if you prefer a chair, there is a room right over there that has the presentations up. If you have questions, we'll have associates in that room as well to try to grab you. So we encourage you to go grab a chair in the other room as well. It's just right here. Sorry.
Can you have me? Laser point?
Yeah. Just right
I
here.
think she needs a laser point.
I got it.
Yes. Yes. Thank you. Good morning, everybody. My name is Michela Locci. I'm an associate professor at the University of Pennsylvania, and I'm really excited to be here today to tell you about our endeavor in targeting the cytokine activin A for the treatment of rheumatoid arthritis. So, first of all, I'll start with my team. That's composed mainly by myself. I'm an immunologist with over 15 years of experience in the study of regulation of antibodies by T cells. Other members of the team are Diana Castano, senior staff scientist with experience in autoimmunity, and two very talented technicians. So we are moving the first steps at the moment to set up our company that will be named Acturion Bio. And this company, the goal of this company will be to harness activin A targeting biologics for the treatment of autoimmune diseases. And today I'm going to tell you about our proof of principle study on rheumatoid arthritis or RA. So RA is a very complex and heterogeneous autoimmune disease, and at the moment there are multiple options for drugs. They are divided roughly in 3 categories. So there are the canonical and targeted synthetic DMARDs, which stands for disease-modifying anti-rheumatic drugs. And then there are biological DMARDs, such as TNF or a strategy aimed at targeting B cells. However, they have a couple of major caveats. The first one is that, for the most part, they tend to cause fairly broad immunosuppression. And some, more than others, they tend to have significant adverse events. The second caveat is that their efficacy is limited. So if you think about methotrexate, which is a first-line treatment, it only works in 20 to 40% of the cases. And the biologics individually can drive remission in roughly 20% of the cases, and they can stop working. So resistance to the drug can be developed. So now the combination of these different DMARDs can keep under control roughly 70-75% of RA cases, but there is still a significant fraction of RA patients that do not respond to the currently available treatment. So there is a major unmet clinical need and an opportunity to develop a new class of therapeutics that works to more precisely target only the undesired immune responses instead of targeting immune responses broadly, and also to be safely tolerated. So the way we plan to do that is to target a type of T cells that regulate antibody responses. So these B-cell helpers are called T follicular helper cells, or TFH, and I've been studying these cells for almost 15 years now, so we are expert in the field. And what these cells do in lymphoid tissue is that they provide helper molecules to B cells in order for them to differentiate into antibody-secreting cells. So usually TFH are tightly regulated, but they become dysregulated in autoimmunity. And that's a problem because they are present at very elevated numbers, they give too much help, and this can result in the production of autoreactive antibodies that then in turn cause tissue damage. So we plan to target TFH, and I want to highlight that none of the existing DMARDs specifically target TFH cells. How do we plan to do that? We plan to target the cytokine activin A. So this cytokine is a cytokine that belongs to the TGF-beta superfamily, and it's way understudied in comparison to TGF-beta. And my lab is one of the few labs around the world doing high-profile work on activin A. And during my postdoctoral training, I discovered that activin A is one of the most potent inducer of TFH differentiation. So now, interestingly enough, if you look at the levels of activin A in different autoimmune diseases, you find that it can be very elevated. That's the case for RA, and it's been published in multiple papers. And this is actually our own data. So we validated the presence of high levels of activin A in the serum of RA patients in comparison to healthy donors. So we develop our own activin A blocking antibody. For now, we are calling it with the clone name, 1051. We know it's highly efficient and neutralizing activin A in vitro. And then we wanted to test it in vivo in an array mouse model called CIA. So it's a model where you drive arthritis-like symptoms by immunizing twice with collagen plus strong adjuvant. So the mice develop anti-collagen antibody that drive the disease. So we first administer 1051 in a prophylactic fashion, so starting the administration at the beginning of the immunization as a proof of principle. And what we found was fairly striking because, as you can see, there was a massive reduction in the incidence of RA and in the severity of the RA in the mice that receive our 1051 antibody. But of course, you might argue that people go to the doctor when they're sick and they have symptoms. And so an important question to us was, does 1051 works even therapeutically? The answer is yes. So in this study, we use, again, the CIA mouse model, and we start administration of 1051 when the mice were already presenting a mild disease. And as you can appreciate, after a few days of treatment, the mice receiving 1051, their phenotype stabilize, whereas the control group go on and develop a more severe disease. So 1051 prevents severe arthritis in mice with mild disease. What about the mechanism? So our hypothesis was that blockade of activin A was going to result in decreased differentiation of TFH cells, and that's exactly the case. So we see reduced frequency and number of TFH cells. Interestingly, we also uncover a secondary mechanism, which we were not expecting, and we did that using a slightly different model that is called Kaya. So in this model, you transfer to induce the disease a commercially available cocktail of anti-collagen antibodies. So the antibodies are present, you are not regulating their production. And after the transfer of this antibody, when you give an insult, such as LPS, the mice become rapidly sick. So now, when we give 10E51 in the setting of this Gaia model, you can see that also in this case there is a significant reduction in the severity of the disease, meaning that 10E51 also prevents arthritis when the autoantibodies are already present. So going back to our model, in line with our hypothesis, we found that blocking activin A results in a decrease of TFH cells, and I didn't show you this data, but it also affects the production of autoantibodies. There are less pathogenic antibodies that are produced. And also importantly, it works, blocking activin A works even downstream of the antibody production by preventing the pathogenic effect of the antibodies. So now, I show you that the advantage of our approach is to deploy a new mechanism of action, so targeting the B-cell helpers, and the preclinical data show high efficacy, so it's a very promising approach. We also believe it's a safe approach. We do not have clinical data yet, but Regeneron has a similar antibody that is currently being tested in phase 3 trials for fibrodysplasia ossificans progressiva. So this is a genetic disease, it's not an autoimmune disease, but the important thing is that results from phase 1 and phase 2 with this antibody show a favorable safety profile of this antibody. And lastly, I want to highlight this is a platform technology because, as you know, there are many autoimmune diseases characterized by autoantibody production, and several of these have already been reported to have elevated levels of activin A, including systemic lupus erythematosus, or SLE, multiple sclerosis, and myasthenia gravis, which is an orphan disease, which can make things even potentially easier for approval of this pathway, and potentially many others. In terms of commercialization opportunity, just thinking about RA, for which we have the most data at the moment, RA is a very common autoimmune disease, if not the most common. So the incidence is very high, can be up to 1%, and there are more than 17 million people affected globally by this disease. So the market is huge. It's around $30 billion for last year. And interestingly, epidemiologic data show that the incidence of RA is increasing. So this means the market will grow further in the next decade. And finally, just to highlight the developmental path, we are now in stage 1. We have filed the IP provisional patent last week. We receive, for a second year, funding from the Colton Center to continue our mechanistic work in the setting of RA, and also to assess efficacy of 10E51 in mouse model for lupus. What's coming next? Still in phase 1, as I mentioned, now we're moving the first step to set up Acturion Bio, and I'm actively looking for a partner, a co-founder, somebody with expertise in antibody bioengineering, and also a CEO with expertise in pharma. I'm also working towards securing funding for stage 2 and 3, not only NIH, but also private investor, specifically those that are looking for a diamond in the rough and that are excited to share this journey with us. Next year, we plan to humanize the antibody. Our antibody is a mouse anti-mouse, but activin A is 100% conserved across species. So the Fab doesn't need to be replaced, only the FC. Perform PKPD, start GMP production with the idea in year 3 to start clinical trial and expand this platform to additional autoimmune diseases. So sorry for being late. I'm going to stop here with my contact info and acknowledging my funding. Thank you.
That's great.
Thank you.
Is your antibody depleting? That's my first question. And the second is, how does this antibody compare to other TFH, targeted agent like IL-21 and ICOS? Okay.
Yeah. So the question is our antibody? How does it compare with other approaches for depleting TFH cells, such as ICOS? And sorry, what was the first question?
Whether it's depleting.
Whether it's depleting. No, it's not depleting, it's neutralizing. It blocks activin A. It works very, very nicely in vitro with the reporter cell line to prevent binding to the receptor. So we haven't done side-by-side comparison, but I believe that, for instance, in comparison to ICOS targeting approach, our approach is more specific because ICOS, it's a costimulatory pathway that may be required also for additional cell type, whereas so far we think that targeting activin A might be more specific to TFH cells versus other T cells.
Yes, yes.
The Regeneron antibody, have you evaluated that, and how does that compare with your molecule?
We just began to do that. So in vivo is a little bit complicated because the Regeneron antibody is a fully human antibody. So there is an FC problem of potentially rejection, so we can't use it for long-term studies. We have tested, we have compared the IC50 for neutralization, and it's roughly comparable with the Regeneron antibody. The Regeneron seems to be maybe a teeny tiny better, but they're the very same order of magnitude.
Yes.
So now that you've got these very interesting results, what will stop Regeneron from taking their antibody and now testing it in RA?
From taking our antibody or their antibody? Well,
Being way ahead of you, by the way.
well, I mean,
You've done phase 1 and phase 2 in another disorder.
yeah, I mean, that's true. No, I guess the answer is that nobody can stop them, but I feel like I have accumulated at this point so much expertise in the study of TFH cells and in knowing activin A pathway that I feel like this gives me an advantage in deciding what the next steps are.
I think that's all the time we have. Just to keep track of time.
Yeah, what's the effect on vaccination, on conventional immune responses?
So we're doing the work with knockout mice lacking the receptor, and it looks like when you have a viral infection, like with LCMV or flu, the responses are largely normal.
Yeah.
So that's as far as we went.
Dr. Locci trained in the lab that discovered one of the labs that discovered TFH cells. So she does have extensive experience in this area.
Thank you.
Introduce yourself, Jim.
Okay, all right.
Do you want a laser pointer?
Yes. So how should I scroll over? Got it. All right. It's my great pleasure to be here. Thanks, Joe, and thanks, Makoto. My name is Jin Wang. I'm assistant professor at NYU. Today, I'm excited to share with you my research programs on key immune feedback modulators as a next generation immunotherapy for cancer and autoimmune disease. This is my team. Includes experienced biotech veterans and leaders in immunotherapy. So myself, besides PI at NYU, I have over 20 years experience in immunotherapy. I'm also founders for Remunix and also founding scientist for NextCure and some other companies. And also, I was lucky to have Leo Slumberg to advise me and also my programs. He's currently a ER at Colton Partners, and also he has been leading drug discoveries for cancer immunotherapy, including ipilimumab, nivolumab, and many others. I also lucky to have some scientific advisors from both cancer and autoimmune disease side, and from Yale and also from NYU, including Joe and also Jill from Colton Center. So what are we thinking about? So we're thinking about unmet medical needs for immunotherapy. First of all, for autoimmune disease, these are very hot areas of studies, and currently, there are antibodies targeting B cells and the CD20 and above, or recently CAR-T and the T-cell engagers has been very interesting. And also another field of study is to block cytokines, cytokine blockade. But however, for myeloid cell and T cells, those are pathogenic populations in the autoimmune disease. We are lacking of effective targets. For cancers, PD-1 has been so effective, but the problem here is for cold tumors, even for some hard tumors which have limited T-cell responses, PD-1 response was very poor. So there is unmet medical needs to identify effective targets for PD-1 in sensitive tumors. So overall, the estimated market value, of course, is over $10 billion per year. So what we have been thinking about, the field has been talking about PD-1 is a magic targets, so effective. Those are immune checkpoints. And recently, there are some PD-1 agonists has been applied, has been showing some efficacy in the clinic, especially in RA.
But unfortunately, many other checkpoints besides PD-1, PD-L1 do not work so well. So what's the problem here? I think it is time to target not just general immune checkpoints, not just another suppressor. It is something in the disease lesion to form immune feedbacks. What I mean is, and also those can be effective for both cancer and autoimmune disease. What I mean is, PD-1, PD-L1 is not just like immunosuppressors. T cell secret, especially PD-1 positive T cell secret interferon and induce PD-L1 ligand. So this receptor ligand suppress T cell responses from lectin feedback in the disease lesion. That is why if you target these pathways, you will recover immunity only in the disease lesion, and this pathway also from lectin feedback loops. That is very critical. So my lab has been very actively looking for other immune feedbacks, lectin feedbacks in the disease lesion, which we can use to boost immune system, repair immune system for in the disease lesion. It might have better efficacy over toxicities. In this way, we can do precision immunotherapy for both cancer, autoimmunity, and other human disease. So we have established the whole platform. Basically, we have the priority functional screening platforms to identify those type of regulators, and also have informatics to identify feedback modulators, feedback finders in the disease. And we have been working on receptor ligand discoveries, identify some key molecules in the interface of disease and immune cells. And we have been very actively looking for the pathway biology of each pathway's molecules for their therapeutic development and also efficacy and the translation into clinic and therapeutics. So the first target we're thinking about is focus on type 1 interferon, because this is a key pathway, critical pathway bridging innate and adaptive immunity. This is the molecules induced by STING, DNAs and pathogens induces cytokines, and ultimately important for anti-tumor immunity, antiviral immunity, and also for autoimmune disease protection. So most of those type of interferon has been produced by myeloid cells. So historically, recombinant type interferon has been applied for multiple disease, and because this is a systemic injection, you have a quite very weak efficacy, but you still have quite significant toxicities. So what we have been thinking about is identify key feedbacks in the disease, inflammatory site, and then this pathway can be triggered by STING or inflammation, and then shut down type 1 interferon production. Because this is immune feedbacks, we can use antibody blockade to reverse that. So we can use this type of molecules to recover or immunity and only in the localized inflammatory site. That is what we have been thinking about. So the first application is multiple sclerosis. As we know, type interferon has been proved for multiple sclerosis. Why type interferon is therapeutic? People are thinking about type interferon can modulate immune microenvironment, especially immunosuppressive macrophages to suppress B cell, T cell, and even type interferon have antiviral function to shut down, like EBV. And type interferon also have some function to protect neuron. That is something quite interesting. So type interferon has been approved, but in general, it's not very efficacious, and 30% relapse in 2 years, and the high toxicity profile. That is a problem here. And then recently, B cell targeting CD20 has been proved is better than type interferon, but only in relapsing remitting MS. So there are other type of MS, for example, progressive MS, anti-CD20, many of the drugs did not work very well. There's a huge unmet medical needs, 1 million patients in the US, maybe 40 billion projected market sale, per year. So this molecule is very new, discovered by my lab. So this molecule is very specific, expressed on myeloid cells, not T cell, not NK cell, DC, and macrophages. And this molecule, if we have knockout mice, first of all, also very conserved among human and the mouse. In the knockout mice, macrophages, we found profound enhancement of NF-κB alpha-3 pathway, which produce type interferon. At the same time, if we overexpress this molecule in Jurkat cell, which normally do not express these molecules, you will shut down Jurkat NF-κB pathway very potently, even more potently than PD-1. So our antibody can reverse that process. This is an antagonist. At the same time, so we found in macrophages, if we knock out these molecules, you have much more enhanced type interferon production. This is a protein level. What's even more interesting is we found STING agonist or even toll-like receptors can induce these molecules. because those molecules are important for tapering production. That is why these molecules induced by inflammation from negative feedbacks make sure those type of targeting will be localized in the disease. So in MS model, which is EAE model, we found our antibodies, blocking antibodies, can be therapeutic. Injecting as day 1, which is prevention, injecting at day 12, which is therapeutic. Both regimen will be very effective. So we look into the spinal cord, we found much less inflammation, and actually we also found some evidence of demyelination, which is linked to progressive MS. So we do think this is beyond just controlling inflammation. And other disease, for example, Burkitt disease or some other autoimmune disease, can be also controlled by interferon, which can be interesting for these therapeutics. So on another side, cancer, anti-interferon also being approved, but it's not very efficacious. We found our antibody as a single agent. It is very effective as a monotherapy. And this is dependent on anti-interferon because interferon blockade completely wipe out the difference. And also, if we combine with PD-1, all the mice survived, no tumor at all. This is a hard tumor. In cold tumors, even more interesting because monotherapy is better than my best PD-1 antibody and the best LAG-3 antibodies combination. So single agent is better than two molecules together. And this molecule also showing quite interesting correlation with cancer survival and also PD-1 therapies. So that's a pipeline. We think we can move this forward as autoimmune disease as a first indication and potentially for cancer. And also we have a hope discovery engine to identify other molecules and also pathways. So in summary, we are targeting not just general immune checkpoints, we're targeting immune feedbacks. Our antibody is also cross-reactive to human and mouse. We can humanize right away and have exciting commercialization opportunities for cancer and autoimmune disease. Thank you so much and looking forward to your comments. Yep.
So, thank you. There was a time when the 3 beta interferons in the market were the only medicines for modulating multiple sclerosis. Today, there's 3, 4, 5 different ones. Do you know, would this approach apply to other non-interferon immune modulators for multiple sclerosis? Because beta interferons don't have the market share they used to have.
Yeah, that's a very good point. So that's why in the beginning, we think this molecule only controls type interferon. But recently, we have quite exciting data showing beyond hyperinterferon, especially for progressive MS, immune control, demyelination, or remyelination. That is a new biology where we think can be very exciting beyond just myeloid type interferon.
In your second last slide, you quickly went over bispecifics. It wasn't clear to me what bispecifics you were proposing?
Yeah, so because we are talking about different disease, right? For example, for cancer, right? We are thinking about, because this is a myeloid target, we might make a bispecific with PD-L1 or other, myeloid-driven, checkpoint blockade. For autoimmune disease, because this control shut down type interferon and also potentially other cytokines, we're thinking about, for example, for MS, we're thinking about some other molecules. TNF-alpha or even VLA4, which has been approved for MS, maybe CD40 ligand, which have some interesting indications, also expressed on myeloid. So we're thinking about some synergistic, efficacy potentially bispecific can bring.
Do you know in the cold tumors if you showed the combination with PD-1, PD-L1 blockade having enhanced efficacy?
Yes.
Do you know in cold tumors if it increases neoepitope expression? That would be the idea.
Because that's a very interesting question. We also found knockout these genes or blockade, the macrophage can switch to M1, and also you have more antigen presentation, more class 1 expression. We do think it can reshape the environment to be more immunogenic, control antigen presentation, or many others.
And do you anticipate if you use the combination that toxicity would go up with checkpoint inhibitors in terms of in terms of immune response adverse effects?
That's a very interesting question. So in general, we found our approach is quite safe because expression in the peripheral is very low, inducing in the disease. So far we didn't see any toxicities, but in human or under some condition, that's definitely some kind of something we should closely look at. But so far, we think this approach compared to STING agonist, compared to type interferon recombinant, right? So that's a very different class of therapeutics.
Other questions for Dr. Wang? Okay, thank you.
Thank you so much.
As a reminder, the overflow room is just to the left, or your right.
Shall I? Okay. Good morning, everybody. My name is James Hansen, and I am a radiation oncologist lost in autoimmunity. And here I am to tell you an exciting tale about how I stumbled into a weird anti-DNA antibody that actually we think is about 3 years and perhaps $7 million away from a phase 2 clinical trial in ANCA vasculitis. So quick, brief overview. We're looking at neutrophils and NETs, neutrophil extracellular traps. They're great, but they're also bad, and we need a way to shut them down. We have an antibody called deoximab that is an anti-DNA antibody that penetrates neutrophil nuclei, binds DNA, and inhibits netosis. It works in vitro and in vivo, and it doesn't do anything else bad to the neutrophils. I was trying to develop that antibody for use in cancer, and I spent a long time on that, so there's a huge backlog of data and safety things that has significantly de-risked this asset. And now, with this new finding in netosis, we're ready to pivot into a new opportunity for a new Yale venture that we are calling Nucleocon. All right, so what on earth is he talking about again? White blood cells. Neutrophils are the most abundant, and we've all heard about those for a long time, but over the last 15 years or so, it's been found that they have this other function, which is really weird, really cool, and really dangerous. And that is that when stimulated, they can intentionally choose to decondense their own DNA, dissolve their nuclear envelope, and spit out their own guts, spit out their own DNA and proteins to form this sticky web called a neutrophil extracellular trap. Now, again, that's great when it comes to fighting off infections because those webs are sticky. Bugs get stuck. But when that happens out of control, that can lead to all kinds of disease processes all over the place. And these nets are not just a marker of damage and inflammation. They actually are drivers of disease, like, for example, ANCA vasculitis. So in modern medicine, we now know we would really benefit from a safe way to shut down netosis when necessary. But that just does not really exist yet. So then let's pivot back to my buddy, deoximab. What is deoximab? It is a re-engineered lupus anti-DNA antibody. So it loves DNA. And we have been historically using it because it finds tumors that are highly necrotic, releasing a lot of DNA into the environment so that it can target tumors independent of surface antigens. And what makes it even cooler is it can cross live cell membranes and get into the nucleus of live cells. And it does that through a nucleoside salvage pathway. In fact, it does that in neutrophils as well. And when it gets into the neutrophil, as I will show you, it binds the DNA and it shuts down netosis. So again, just as I mentioned, deoximab likes DNA. It specifically loves nucleosides. It binds to nucleosides outside of cells and then gets sucked up by live cells in the area by an ENT2 nucleoside salvage transporter. That is different than any other antibody that I know of. It avoids endosomes and lysosomes. It gets straight in the nucleus without being degraded. But it can only do that if the DNA nucleoside substrates are present outside the cell. So this is not an antibody that's going to go everywhere. It's only going to find the areas that are enriched in DNA and then penetrate those cells in the local area. And again, I had based pretty much my entire scientific career on the idea of this antibody will find tumors, get in the nucleus, and perhaps we can deliver things there. And it wasn't until 3, maybe 4 years ago, a friend of mine said, "You ever heard of NETs?" And I said, "No, I have not." And he said, "Well, maybe you should, because I think your antibody might have something to do with those." So I looked them up and found out what NETs are, and suddenly this idea of a DNA-rich environment does seem indeed perfect for deoximab. So the first question we had was, will deoximab penetrate a neutrophil? Yep, it does. Even in that funky trilobed nucleus, it penetrates and finds just the nucleus. But then what happens if you tell those neutrophils, go ahead now, release your nets, throw PMA at them? They can't. So you see those nets in the middle? They're not getting released when deoxymab is already in there. You can see that under confocal microscopy. You can see that by Western, showing that the citrulinated proteins are retained. If you want to take a close look at the nuclear membrane, that nice red circle of lamin B1 in the happy neutrophils, if you throw PMA in there to induce netosis, that red circle dissolves. If you throw a control antibody on, doesn't matter, it dissolves. But if you throw deoxymab on there first, no matter what you throw at it, that membrane stays intact. It's not just us that have seen this. This has been now confirmed around the world. We have a great colleague at Monash University, Kim O'Sullivan, who has said, yeah, you're not lying. And in fact, everything that we throw at it, PMA, ionomycin, LPS, TNF-alpha, this blocks netosis of all pathways. Closer to home, through the work with the Colton Center, has helped create a great bridge between my lab and the Cefic and Flavel labs here, who are working with SARS-CoV-2 viruses and netosis. And they said, well, maybe could we try your antibody? Why not? Indeed, deoxymab prevents netosis induced by virus as well. So it's all looking pretty compelling, but you might say, well, this is just killing neutrophils? No, it's not. It's actually making the neutrophils happier. Deoxymab promotes survival of neutrophils even when exposed to toxic stimuli, and it does not impair other functions like phagocytosis, reactive oxygen burst, chemotaxis. It's remarkable. It can penetrate the nucleus, shut down netosis, but so far has not done anything else in terms of interfering with neutrophil function. So at this point we said, well, that's kind of compelling. Where would we want to start for a first indication? And we worked again with the Colton Center, said, you know, the most classic net-driven disease is this disease ANCA-associated vasculitis. These patients develop autoantibodies against neutrophil antigens. They're called ANCAs. The ANCAs bind neutrophils, induce netosis, and those nets drive endothelial damage, vasculitis. There's no therapy that can target that specifically yet. We need something to go after that. So we went after that in a mouse model. It's a very easy model. You just immunize the mice with markers of those neutrophils, and eventually they will induce those ANCA antibodies. Once you've got a good solid ANCA titer, you know the kidneys are in trouble. Give them a dose of a control or the deoxymab, and then check the urine, check the kidneys, and see what's going on. Urine shows good results. White blood cells are down, protein is down, decreased proteinuria, easy things to translate to clinical trial tests. Kidney biopsies, the nets are no longer there. You can't see them in the kidneys anymore. The glomeruli, they look happy, no more nephritis. No more neutrophils, T cells, macrophages, and such. It's a remarkable response. Okay. I mentioned that we have great access here to the Flavel and Cefic labs, who also work in very special humanized mouse models and are in particular interest in COVID-19 and SARS-CoV-2. And who wouldn't want to collaborate with a team like that? And so they said, let's test your antibody in our humanized mouse models. So you take your humanized mice, you express the receptor for the virus, give them SARS-CoV-2, and afterwards, give them a dose of a control antibody or deoxymab, and then take their blood and lungs and look for nets, look for pneumonitis. And again, nets are down in the blood, they are down in the lungs, and the lungs look happier. So it's looking very promising now in 2 different models. Again, there's really nothing out there that can target nets the way deoxymab seems to be able to do so. It's DNA specific, it penetrates the nucleus, it inhibits netosis, and it doesn't do anything else bad to the neutrophils that we've found so far. And again, deoxymab was already pretty much ready to go for clinical trials in cancer. We had a fragment that can cross the blood-brain barrier that passed all the way through toxicology in rodents and primates. We've got GMP and the technical know-how and the IP in place for protection through 2044 in all major markets. And this sudden opportunity to pivot to a new venture, I think, is exciting. And we've got a whole team ready to go, including myself, Jiangbing Zhao, Richard Flavel, and Essen Cefic as co-founders, and a great business and scientific advisory team. So with that, looks like I was able to speed through just on time. We have an estimated 3-year path to getting to IND filing, and we're looking to raise about $4 million to cover the costs of getting it to there through the engineering run and the toxicology and such. And then we think we have a green light to move straight forward to a phase 1/2 in ANCA vasculitis. And if we can get to that point, it would probably cost us about $3 million to get us there. The market is estimated to be about $1 billion in the orphan disease ANCA vasculitis, but of course, NETs don't just contribute to that disease. You move beyond that to NET-driven kidney disease like acute tubular necrosis, cisplatin-induced nephrotoxicity, other things, or even beyond that to viral-induced inflammatory disease, and beyond, the market starts to rise. So overall, this is nowhere near where I expect it to be at this point in my career, but I wouldn't be anywhere else. I think this is an amazing antibody and an amazing opportunity, and I'm really grateful for your attention and happy to take any questions.
Questions for Dr. Hansen?
Hi. So based on what you know, if you were to forecast what the phase 3 trial would be, how much of a reduction in the endpoint for ANCA-associated vasculitis would you imagine this could have?
It looks remarkably effective. So I would envision the deoxymab therapy to be sort of like a Keytruda dosing profile where when the disease is in its flare, an infusion every 3 weeks would knock it down to suppress it. I don't envision this to be a lifelong therapy, only when an as-needed approach. But based on the mouse data, I'm expecting greater than 80% reduction in the markers of the urinary protein in white blood cells. Pure guess.
Other questions? How would you compare, James, just thinking ahead in relationship to that question, the current therapies for ANCA-associated vasculitis, which is largely based upon glucocorticoids, which have significant toxicity, and anti-CD20, rituximab, which also has significant potential toxicity, or complement inhibitors, which haven't really been shown alone to be that effective?
Right, and that's the real big window of opportunity that I think is here, which is that if everything continues along this path, deoxymab is able to hit the central problem specifically of the netosis without causing a lot of immunosuppression otherwise. And that, I think, would be the huge benefit for patients with ANCA vasculitis and hopefully other diseases as well.
A related question, I was intrigued by the idea that there doesn't seem to be much of an off-target effect against normal intact neutrophils versus NETs, which is a really very nice finding, if I may say so. How do you just tell me how that works? And it wasn't clear to me how that works.
Well, so it's not that it doesn't have an effect on intact neutrophils, right? It's indeed penetrating the neutrophils and protecting them and inhibiting the release. But that actually does track with what we saw in the oncology space. Is remarkably, this antibody penetrates the nucleus of normal cells and doesn't kill them or have any adverse effects. But what it does do is inhibit DNA repair processes, which makes them have an effect on vulnerable cancer cells. So when you want to combine things with DNA-damaging therapy like radiation or chemotherapy, your antibody has a good effect on that. But in the absence of those DNA-damaging agents, the antibody has no effect at all.
Other questions for Dr. Hansen? Okay. James, thank you very much.
Thank you.
Okay, thank you very much for the opportunity to speak, Joe, and the support of the Colton Foundation. I'm going to tell you about a company we launched out of Colton funding in December. That's a targeted company that's using applied human genetics to develop autoimmune drugs. The company was founded with seed funding from Nova Holdings. The partner there, lead senior partner, is Jan Peterson. Jan's background is in 40 years in biotech. So he was the CEO and developed Zealand Pharma through exit, and then he became the director of immune disease at Merck for 10 years. Then he moved to Novo Nordisk, where he was the director of immune disease at Novo. And then he moved to Novo Holdings, where he's been a senior partner for a number of years and started a number of companies and had good exits. I'm at Yale. I'm a pathologist, and I actually know Taku. Hi, Taku. It's nice to see you. Taku is also in the pathology department at UPenn. So I read HemePath 1 week a month, and then the rest of my time is spent running a lab. Of interest is that I'm the scientific founder of Inozyme. It was a rare disease company with an enzyme replacement strategy. It announced positive phase 3 data 2 weeks ago and was acquired about a week ago by BioMarin. And now ready to move on here. So Dipender Gill, we're joined. Dipender is from Oxford University, and he worked at Novo and set up the bioinformatics group that validated drug targets for Novo for a number of years using GWAS approaches that we'll talk about briefly. And we're using Dipender's technology in validating drug targets and my understanding of the engineering of biologics now to develop drugs. So the company is envisioned to have a footprint here in New Haven with funding through Novo to my lab, and we're developing the lead asset. The C-suite will be centered in Copenhagen, and then when we move into clinical trials, there'll be a footprint in Boston to access the U.S. markets. It's a multi-asset company. I'm only going to be able to tell you about the lead asset, which is a project that is targeted against lupus. What we are seeking is to raise an additional $30 million to get through phase 1A trials for a first-in-class biologic, and then have the second-line project available for drug candidate nomination. So the lead indication is lupus, and briefly to just give a highlight of lupus, it's one of the leading causes of death of young women in the U.S. This is from recent data examining autopsy and death reports in the U.S. It's a top 20 cause of death of young Caucasian women, and if you're an African American or Hispanic woman, it's a top five cause of death. So it's currently severe unmet medical need. The lead treatment is a non-specific therapy that was originally developed for malaria, on which you layer anti-B cell therapies and suppressive therapies that are toxic. The hallmark of the disease is the development of anti-double-stranded DNA antibodies. These are antibodies to chromatin. When you layer lupus patient plasma onto Hep G2 cells, you light up their nuclei. They're reacting to chromatin. And of interest is that recently, now in the last 3 years, we have identified from two groups, one from NYU, the other from Hopkins, neutralizing autoantibodies against the very enzymes that degrade this antigen or chromatin in your blood. So now this is one of the learnings that we're going to take as we move forward with understanding the disease. This is taken from a review article 10 years ago from an investigator who's an expert in the autoantibodies in lupus. And to illustrate how lupus is envisioned to work, it starts, I don't even know if I can use this, I can't, okay. It starts with the deposition of necrotic cellular debris in your blood. So inhibiting NETs is not going to take care of necrosis and cell debris that's going to now trigger a neutrophilic immune innate response to these sites. The neutrophils are going to try and phagocytose it. If they can't, they're going to explode, reducing more chromatin and DNA into the blood. And this will accumulate, and if it's not efficiently cleared, it's going to be taken up by T helper cells in the dendritic centers, presented to B cells, which will then go through their VDJ recombinant steps, and those with efficient antibodies will immortalize, migrate out of the germinal centers, and become your long-lived immune cells. These will continue to extrude autoantibodies against chromatin as long as it's there. These can deposit in the kidneys, causing lupus nephritis, cross-react with brain antigens causing lupus encephalitis. And now we know from two independent groups that these antibodies also can neutralize the very enzymes that are charged with degrading the chromatin in the blood. And those enzymes are called DNase 1L3. So I got started in this about 5 years ago from this report in rare patients with lupus. This is a pediatric population. They don't have the enzyme. They have a truncation mutation near the N-terminus. They don't make it. They all get lupus. Fully penetrant, Mendelian inherited recessive disorder. Really speaks to disease pathogenesis. 6 years later, reports invoking a SNP in the same enzyme that reduces the expression in the blood by 80%. These patients are now at risk for lupus nephritis, as well as rheumatoid arthritis, hundreds of thousands on the slide there, and scleroderma. And finally, as we mentioned, these now neutralizing autoantibodies in the general lupus population that are present in 30% of patients with lupus and 50% with lupus nephritis. So now, using this information, we developed an enzyme therapy for this disease. And as I went out and raised money and pitching, what they did at Novo was they took the center SNP, which I call a mutation, and they call an instrument for drug validation. And you can run this through a GWAS study in a Mendelian randomized process to develop not association but causality. I want to be very clear. This is not enzyme replacement. We are now going to identify absence or this enzyme with causality in lupus patients. When you run those protocols, it associates very strongly with disease causality, underscoring that this is a drug target that's valid against all lupus patients. So that is how we are going to now treat the disease in a mouse model. We're going to use the standard mouse model for lupus, which is when you challenge the mouse with a chemical called pristane. It induces cellular necrosis in the blood. We're going to hold dosing till the first animal dies. 2 different C57 Black 6 strains. We recover animals from death. If we look at the middle panel, what's driving death in the animals? It's the lack of 1L3 in a knockout animal, not the lack of DNase 1. All right, and so we can recover those animals by doing enzyme replacement, of course. If we look at the disease pathogenesis, the animals die like COVID animals die, like ankylosing animals die, like severe lupus dies. They bleed into their lungs. They leak surfactant into their blood, they lose kidney function, elevate creatinine, elevate myeloperoxidase enzymes, and lose oxygenation. So these are all mechanisms of death common with all autoimmune phenomenon, whether it be lupus, COVID, ANCA, and others. We don't really have time to go into the specific drug, but this and much other data is published now in JCI Insights in this paper in July, including the fact that this drug does not cross-react with the neutralizing autoantibodies in the patients of lupus plasma. We sent the drug to Hopkins, to Philippe Andrade's lab, who has these autoantibodies. 99 pooled plasma of lupus patients, 33 of which had neutralizing autoantibodies, did not cross-react with the enzyme fully active. So I will refer you to that paper. I'm just going to now overview where we're at with the company. So we founded the company, I think it was December 6th, after a lot of excitement here at Yale and pain, but thanks to David Lewin and people in the development office, we got that done. They put $5 million into the company. They're going to put another $5 million in June. So they'll have put $10 million in. We've started manufacturing at Lonza with 6 assets that will go through immunogenicity testing, toxicity, and PK/PD. You'll have full access to all of that data before you make the investment. The drug candidate will be nominated in July. We would like the final, the syndicate to be formed by the end of July for closing in September. Along with that money, then, we are going to take the program through phase 1B, and we will nominate a second program drug candidate in April of 2027, according to this timeline. So this is kind of the next steps looking forward. This program is out of Yale and Denmark, and I know I had to go through with this pretty fast, so I apologize for that. Happy to take any questions. Thank you.
Questions for Dr. Braddock? Anyone? So, Demetrius.
Sofinova had 45 questions for us that we have to answer by next week.
These are not pre-planned questions, I can assure you. These are off the top of the head. It may become apparent that they are off the top of the head. But Genentech had a DNase-1
Yeah.
inhibitor that failed.
Yes.
So maybe it would be important
Yeah.
to explain the differences again. I think
Thank
that's
you.
an important
No,
mechanism.
it's a great point. So Genentech had DNase-1 program. It's pulmozyme. So we have a 20-year experience of DNase-1 in humans. It's completely non-toxic. Their DNase-1 does not degrade chromatin. It does not degrade histone-bound DNA. It degrades free DNA. And as we saw with the mouse studies and also the data in the genetics, DNase-1 as an instrument does not give a signal in lupus. So you really need to have an enzyme that degrades chromatin-bound, protein-bound DNA. Now, our enzyme and our drug is a DNase-1 drug. And that's important because we took that drug and we evolved it chemically into a DNase-1 enzyme to give it full DNase activity, and that's why it shows efficacy. But importantly, Genentech's enzyme was not bioavailable. They just injected naked DNase-1 into the body. It's going to be cleared in first-pass metabolism, and it had a half-life of 30 minutes in patients. Now, they took that enzyme and they dosed lupus patients, and there's a publication in 1999 out of the NIH in which they took DNase-1, they gave it IV bolus into lupus patients and then 10 sub-Q injections, and they found the enzyme did not elicit autoantibodies in those lupus patients and was completely safe. It was ineffective because it was not bioavailable. And also now we believe it's ineffective because it doesn't degrade chromatin. So on that backbone, however, we've built a drug that we already have insight into as being safe and non-immunogenic in lupus patients. And so that's a significant advantage as we move forward with this program because we already have some experience with humans with the scaffold of the drug backbone.
Thanks, Demetrius. I think that's helpful. Yes. Yes.
I'm struck by your statement that you have found the cause, the underlying cause, not correlation.
That's exactly right, yeah.
And
it's a strong statement, I agree.
Strong statement. And so many therapies for autoimmune diseases in the past have gotten approval without any understanding of the pathophysiology of how the disease works.
Yeah.
So how does that fact change the responses you're getting from potential funders? Does it move them to understand the disease?
Well, there's some that remain unconvinced, certainly. Some even in this room, I believe, who remain unconvinced. And then there are some who are very convinced. And so I think we've pitched to 50 investors, and we have 11 in the data room, and we've had follow-on calls with substantial investors. I have to say that while I was trying to raise this first round, it was difficult, and the key for Nova was running these Mendelian randomization algorithms, which they strongly believe in. So I think it's either something that you really believe in or you don't, and it's used now routinely in the pharma industry, and it's not my expertise, frankly. So. I guess those are my comments. There's strong uptake. We think we'll be able to close the round, but certainly there's always going to be skepticism.
Other questions for Dr. Braddock? Okay. Thank you, Demetrius.
Thanks.
Hi, I'm going to introduce myself today. I'm working with Dr. David Pitt. He's at the Yale School of Medicine. He's right there with his postdoc, John Guerra-Cody. He's the director of the National Multiple Sclerosis Brain Bank. I have worked with him for many years. I am a biotech analyst, biotechnologist. I've been involved in many startups. I was head of academic collaborations at Bristol Myers. And I've assisted and helped and identified a lot of in-licensing. I have to put my disclaimer here from Catalyst, which I'm working for now, that Dr. DePaoli is an employee, and this work is unrelated to Catalyst. You can tell it was written by a lawyer because he calls me doctor, which only my mother does. So this is the team. We already sort of started. Dr. Pitt, long experience in multiple sclerosis, as do I. And John, who's the sort of genius behind the data part of this diagnostic tool. Dr. Sue Gels is a very well-known biotechnologist from Biogen, who I also worked with in the past. She's assisted us greatly in putting us together with collaborators for the data that we've collected, this sort of high-quality data we collected. Manuel Moore at Yale Ventures and myself. We are pre-seed diagnostic startup. We sort of operating under a new co-identity. We haven't really formed that yet. But we do have our founder team. So we're doing diagnostics. The talk is largely going to focus on MS, although what we have developed is a platform for many diseases. I'm just going to give a quick background on what MS is. MS is an autoimmune-mediated neurodegenerative disease. Strikes people in their early adulthood, and it has two phases. One is a relapsing phase where patients have attacks. They can remit or recover from these. And this is where almost all the treatments are for MS. At some point, over here it looks like a sharp line, but it's actually a sort of unknown time. Two-thirds of patients will start to progress. Without any attacks, they will have, or between attacks or no attacks, they will decline. And this is really where the most devastating part of the disease is, where there aren't very few, no therapies really, and where the greatest impact on patients' quality of life is. This is where they lose their job. This is where they're unable to participate with their family. They go from a cane, to a walker, to a wheelchair, to bedridden. Now the issue is, we have a hard time diagnosing when a patient is progressive, never mind when they're going to become progressive. So the standard of care is to look backwards with how have you been doing over the past several years with some very sensitive and time-consuming neuro exams. There's some use of MRI, but really it's looking backwards. So it's a retrospective diagnosis. It's mostly believed this is, Hartung says this, the 3 to 6 year delay, maybe longer. Most physicians don't do this test. What we'd like to do is, in the little green dot there, is bring that diagnosis back from this unknown orange triangle to diagnosing people at the beginning or even before they have their progression. And we think we have done this, and I'll show you the data for that. What does that do? So at this decision point, physicians are left with not knowing where their patients are going. They will either leave them untreated, where they lose years of this disability that maybe they could have done a more aggressive treatment and delayed their disease progression, or they overprescribe a drug that their patients don't need, which have serious adverse events because they're now immunosuppressed. So this is really about right drug, right patient, right time. And without a biomarker, you don't have therapies and you don't have treatment decision making. There's a great need. It's important for the clinic. It's important for the development of trials in progressive MS. No trial exists without a good biomarker. And these drugs aren't cheap. Putting a patient on the wrong drug is $200,000 a year per patient. Our strategy has been to look at exosomes. Exosomes are ultra-small membrane vesicles. They're very durable. In this case, we can get them with liquid biopsy from bloodstream. They're not one thing. There are different exosomes released by different cells. We often describe them to people as a package or a letter of sorts where there's a to and a from and addressee, and there are contents, complex contents of nucleic acids and proteins which you can analyze. So it's really if you can pull these up, you sort of pick up where it came from and what the state of the cell that you got it from is, which is really what we're looking at. And not just exosomes, we're looking at particular exosomes from astrocytes. So we focus on astrocytes. These are an important cell for neuronal support in the brain. They're the bulk of the brain. They are the communication between the brain and the periphery. And they're involved in almost every neurological CNS disease. And they're activated in many diseases. Activation of astrocytes is a hallmark of MS and progressive MS. We've pulled out astrocyte exosomes, as I said, it's accessible peripherally. And from this, we get this complex signature. We are not looking at a single biomarker for a diagnostic. This is a family of molecules that give us a really complete picture of the disease pathway, not just something like neurofilament, which is there's some neuronal damage of some sort. And then we apply, in addition to pulling out this biological material, we apply that data to a deep learning model, which John, our postdoc, is in charge of, that sort of brings this as a sort of AI plus bioassay combination. This is the process. I've sort of gone over it. We pull out cells. We isolate astrocyte-specific exosomes. We then take those contents and sort of identify what signatures are involved in different diseases, and then we do a deep learning to combine that with the patient outcomes. And this is our data. The red circle so these are patients we know what their condition is. They're either progressive or long-term stable MS patients. And blue is the stable patients and red is progressive. And these very clearly separate. The area under the curve here is 0.95. This is a remarkable separation for a blood-borne assay that probably is equal or superior to what a physician would do in the clinic. It's superior in that it's in real time, it's not retrospective. And additionally, because we have at Yale, we have a great MS laboratory clinic and community, we have a huge sample database in the brain bank. We can look at patients who we've collected blood from in the past and say, did this patient they're stable today, but did they become progressive 2 years from now, 4 years from now, 5 years from now? And this separates actually even better, the area under the curve, 0.97. It's a smaller number, but and once again, we are able to separate. This is remarkable. This is something no clinician can do, of saying you're going to be progressive 2 years from now or 4 years from now. So this is there's one data set. We've applied this data to many different dimensions. We've done CSF. We have a brain bank. We look at brain samples. So this works. It's not a matter of wondering if it works. We're very close to completing our validation in a number of dimensions. So what is we've sort of gone over the clinical necessity of identifying the diagnosis. This is really our path to profitability also. The first is obviously developing as a clinical diagnostic, either a CLIA test or FDA approval. There are 1 million MS patients in the US. About 680,000 of them are over 45. These are the patients who would need an annual or biannual test. They already come into the clinic once or twice a year for blood draws anyway. Presently, we're charging $1,000 per assay to do this. It's not in the clinic. We're doing this in a trial and research basis. That represents about $1 billion market. The market for biological, I know a lot of the talks today would be on drug development, but the market for biologicals is large, $9 billion. We are also participating in pharma services. We're involved in clinical trials. This allows us to develop our products and in a randomized clinical trial population. This is extremely valuable to us. We are not paying for this. We are being paid for this. It's a source of revenue, but it's also a savings of many tens of millions of dollars in validating our method. About a minute left. We have competitive landscape. I would say there really isn't a competitive landscape because there is no progressive MS biomarker. There's no MS biomarker. There's Quanterix that does neurofilament. They're a $122 million company. Their marker is a single marker. It's not specific to MS. It's just, you can hit your head and get neurofilament. But there's a code for it. You can get it as a clinician, and they make money doing that. I think what we have is vastly superior. Octave is another company. They have a protein panel, which is interesting, but it's really about this relapsing phase. There's really nothing for progression. So I think we're really positioned to be a first to market. I'm not going to go over this in the last minute, but the market for neurological markers is growing, as we've seen for Alzheimer's. Once there's a disease and there's a drug and there's a marker, there's an intense pressure to include this market to treat early and to treat correctly. We have other products involved in our what we're doing. We're involved in trials for this: ALS, aging, long COVID, NMO. This has worked really in everything we've tried, of which MS is our lead candidate. That's our strategy, sorry, going forward. I'll go half a minute. So this is our strategy. As I say, we're in early, our validation is close to completion. We're locking in our IP. We are participating in randomized cohorts, and we're developing our pipeline. In the next 12 months, we'd like to expand our randomized clinical trial validation, secure a CLIA or FDA submission feedback. Diagnostics is not like drug development. You can get this done in a month or a year at most. So we are very close to an area where we're profitable, and we'd like to develop the platform, not just necessarily out-license one test. And that's it. We're developing gold standard.
Thank you.
Thank you.
So questions for Ilir, please?
Yeah, thanks for your presentation. I saw you said you need 0.5 ml of plasma. Can you speak to any sample handling requirements? Do you have special blood collection needs, and then how does it need to be transported
that's
for isolation
answered by
of
the
exome?
by the inventors. But the exosomes are very durable. These samples can be frozen for years. There's a plasma requirement. We're doing 0.5. We're actually working. Part of our goals on here are productization of scaling up, maybe using less volume. Maybe depending more on PCR. Now that we have a signature, we may not need to isolate exosomes. So this could be scaled up, and the cost can be brought down remarkably. And they're very durable. That's the good thing about exosomes.
Very interesting. Where do healthy individuals fall on the PC1, PC2 plot?
We have it. There, I said, David, they'll never look at this data, and here it is. So I think it separates actually pretty well. You can sort of see the healthy patients are great in the upper left. These are all the diseases combined on top of each other. But obviously, you would know something about a patient before you sent them in. But healthy controls separate differently than
So the MS stable are more close to the healthies.
Are closer to which you would expect, right? They don't not having any less active immunity. They're not normal, but I think David would know the answer.
We also looked at long COVID because one of our lab neighbors does long COVID, and that's a relatively subtle disease compared to ALS, for example, and you can see they cluster closely together, and progressive MS and ALS, they are further away.
Yeah, you can really see how you can apply to many dimensions of anything that involves the CNS.
A question for treatments and progressive MS, by and large, not so successful. So is your hypothesis that early diagnosis and early initiation of those treatments will have better efficacy, or do we actually need different treatments for these patients?
I personally think it's both. I think, first of all, the trials have been done on what we call progressive patients. A lot of them are not progressive. They're relapsing patients who are just between relapses. I don't think you can create a progressive specific drug with a progressive specific biomarker. We're using relapsing remitting biomarkers for progression. I think that's the problem. So I think there probably is a new mechanism necessary, but there are drugs, Ocrevus, which is where we're thinking, has been shown to delay progression. So that's a dimension of it, and where you would make that treatment decision. Should we put you on Ocrevus? Maybe we delay progression, after which we do need clinical trials to affect progression or not. Right, so it is essential, like any clinical trial in progression needs to use a method like this for patient selection, or maybe for an efficacy outcome.
For your principal components analysis, how about RRMS patients? Can you predict flares versus the remitting stage? What do they land on that?
Relapsing versus remitting?
Well, flares versus
I don't think,
remitting. Or remission?
I don't know. You would have to ask them, but I don't think we would really be able to. We are differentiating that. That's not really the question we've been pursuing, since there are.
I know it's not your question, but I was wondering if you're able to differentiate or predict. Because I'm thinking of, I know you're using this for progressive disease, but even for endpoints or trials.
I don't think we've looked at like that fine a time scale of like the month before and the month after a relapse.
Okay, so you can't predict flares or a patient that's going to flare.
David can answer. He would know.
But we looked at patients who have enhancing lesions, which is a hallmark of
active lesion.
Goes together with a flare, with a clinical symptom. We can distinguish a signature between that and patients, relapsing remitting patients who are stable. We haven't looked into predicting that.
So I guess the answer is yes. Yeah. Shortly, briefly, yes.
Ilir, you talked about the competitive landscape looking at urine serum proteomics.
Yeah.
Of course, that's pretty hot now because of the recent data in Alzheimer's patients.
Correct.
As a predictive marker. So maybe you can expand a little bit on the comparative use of exosomes versus proteomics. That's obviously coming, and there'll be improved platforms and so forth.
I guess it's an exciting field now. Biological markers in neurology are exciting. So I would say they aren't a competitor, but they're sort of a role model for what's happening. None of these trials in Alzheimer's would have worked unless we identified patients who have the target that's necessary. Many of these trials failed. They either didn't have Alzheimer's, right? These are the Pfizer trial failed, 15% of the patients didn't have Alzheimer's. Other ones don't have amyloid, so you're giving an amyloid drug, and this is how Biogen revived their drug trial. They showed that they had target engagement, and the patients who had reduction in amyloid had a response. So I think it's necessary. And then this translates to what we're doing in ALS. If we can identify the right population, then obviously we come out as a companion diagnostic. That in the same way that there were no diagnostics for Alzheimer's. Now that there's a drug, people are doing CSF, then they're doing PET, and now they're doing blood tests. The pressure for technology is immense, and I think that's the case here. But I think we picked a good technology.
And one follow-up question, a follow-up to Dr. Clancy's question. So how long are the exosomes stable in plasma, and how does the plasma have to be handled? And I assume it's plasma, not serum, right? Plasma. So how long are the exosomes stable in plasma at 4 degrees or at room temperature? Some of these are, I know, historical samples that have been biobanked, but in terms of real-world usage.
Yeah, I think I can answer that. So we process the samples as soon as they're thawed. So
Hi, thank you. So we process the plasma samples as soon as they're thawed, and when we isolate platelet-free plasma, we freeze them within the hour. And we don't do freeze-thaw cycles because we've seen that exosomes do rupture when they're frozen, and we lose the yield. So we do it within the hour of thawing, and they're not thawed. They're thawed very slowly to prevent bursting.
We haven't done a timeline also, but we get samples from, for example, from the MS clinic. So a sample is drawn at 8 o'clock in the morning or something like this. It will end up in our lab at noon or in the afternoon, and we see good results with that.
All right, I do have a question here. So thank you for the presentation, very interesting. So other than astrocyte, how about the oligodendrocyte and the microglia? Those can be a biomarker as well?
I think they could be. I mean, we've chosen astrocytes because they're so clearly affected in MS. I think the astrocytes are, you can pick any of these cells, but I think the astrocytes are really a great target. Because
oligodendrocyte can be also linked to progressive.
We're doing this now.
Yeah.
We're looking at
Got it.
exosomes.
Yeah.
There are many questions that you can answer. That remyelination.
Yeah, a microglia is more a hallmark of, you don't have it in healthy patients as well, right? So it's different, the same activity.
Thank you, Ilir. So that ends our session. Thank you to all the speakers and the audience and the questions.